Hello everyone, welcome to part 3 of glycolysis. So just a quick summary, and we're going to go over certain things we talked about. We're going to move into a little more detail. But again, in part 1 we had all 10 steps of glycolysis in detail. In part 2 we looked at a little more of the products and substrates, and today we'll look at pyruvate oxidation as well as the end products. So again, glycolysis takes place in the cell cytoplasm, or the cytosol. So glycolysis is essentially when we split glucose, the yield ATP, producing equivalent NADH to produce ATP via oxidative phosphorylation. Then we produce pyruvate. Then this provides the substrate for phase 2 of glucose oxidation. So again, glycolysis can use glucose or glycogen. So again, the use of glucose or glycogen depends on a fed or fasted state. So again, we've mentioned that glycogen is kind of our resting glucose. So this will be used in our fasted state where glucose is fed. So we have that glucose in our blood from that food. Again, this also applies to rested versus exercise. So as we exercise, we increase the need for glucose. So we might have to go into our glycogen to keep our energy up. Again, glycolysis occurs all the time. The rate of glycolysis depends on ATP, such as rest versus exercise. And again, there are 10 enzymatic reactions, so it's relatively fast. So again, we talked about this figure last lecture with the different energy systems and their timing. So again, immediate energy source is our ATP creatine phosphate. Then we have our glycolysis, occurring in around 10 seconds, which is our short-term energy, and then we have our aerobic, which is our long-term energy. So again, the end product of glycolysis is pyruvate. And here we can see pyruvate right here. So again, pyruvate has 3 carbons, but glucose has 6. So during glycolysis, 2 molecules of pyruvate are produced from 1 mole of glucose. So again, glucose, 2 pyruvates. Just the short form. Pyruvate is found in the body as an anion carrying a negative charge on oxygen following the dissociation from the hydrogen atom. So here we see that negative charge on the oxygen, making it an anion. So pyruvate is essentially a crossroads of metabolism. So there's anaerobic metabolism, which we will talk about at some point. And essentially this is when, if we don't have oxygen available, so sometimes when we exercise at extremely high intensity, like sprinting, we're not getting oxygen fast enough for the energy required. So we have to use other mechanisms like we're going to produce using lactate dehydrogenase, produce lactate. So essentially, we know a lactate, you guys have heard of lactic acid. So essentially, if that sprint where we don't have enough time to do that, or resources, we go and use lactate dehydrogenase, and this is our very quick, and doesn't involve oxygen. But we can see here there's 3 couple pathways. So pyruvate can use lactate dehydrogenase to become lactate, and NADH is used, and then we usually get the NAD plus back. That will go back into glycolysis. We can also have alanine transaminase occur to produce alanine. Pyruvate can also use the enzyme pyruvate carboxylase. So here we can see this requires energy, and CO2 enters. This can get oxaloacetate, which we'll talk about how this can enter this citric acid cycle. And then oxaloacetate can use citrus synthase to become citrate, and then this will go to the citric acid cycle. Or it can use pyruvate dehydrogenase, which is the most common, at least in oxidative metabolism, where we have acetyl-CoA form, we have a CoA join, NAD plus is used, we get an NADH, we produce CO2, and we form acetyl-CoA. Then this acetyl-CoA forms a citrate, forms a citrate synthase, forming citrate, entering the citric acid cycle. So again, there's many different ways for these pathways to occur. It's not exclusive to one pathway, but you can see that pyruvate is very important as a crossroads of metabolism. So its function is essentially crucial. So pyruvate. Phase 2 of glucose oxidation is where we have pyruvate dehydrogenase reaction. So you see pyruvate dehydrogenase is usually discussed as PDH. So again, here we have pyruvate. So if we have no O2, it becomes lactate. So with the presence of oxygen, it enters, pyruvate will become acetyl-CoA, enter the citric acid cycle, producing our carbon dioxide, NADH, which enters the electron transport chain, which then eventually goes into ATP synthase, producing ATP. Then we can also have pyruvate to lactate. So again, we can have our alactate or lactate metabolism. So lactate formation. So lactic acid, so that's our La, is formed continuously through glycolysis. It's largely combusted as fuel. So lactic acid gets a bad rap in this thing with muscle stuff, but this is not necessarily true. It's essentially the hydrogen ions that cause that thing. It's not the lactic acids. So we'll talk about that later, but lactic acid is used in certain things like the Cori cycle, and is used actually in energy. So a lot of athletes that produce a lot of lactic acid or actually can benefit from it and use it as an energy source. So it's not actually this negative thing. The negative thing is essentially the negative charge with these hydrogen ions. But again, we'll get into that in more detail. So lactic formation is essential to generate NAD+ to go into glycolysis at step 6. So without oxygen, at least in this case, we need glycolysis to continue. So we need this NAD+. So essentially, we form that 2 NAD+ and NADH in glycolysis, and this is used during fermentation reaction, and we talked about this in our biology course. So we get our 2 NAD+, and this goes back into glycolysis. So here we have our net 2 ATP. So essentially, you don't get any more energy. It just allows the glycolysis cycle to continue by giving it back its NAD+. So essentially, it's a process to get this. But again, lactic acid can be used as energy as well. So as I said, lactic acid in dissociation to lactate and hydrogen ions causes coincidental acidosis rather than causal. Instead of ATP hydrolysis. So this is that kind of, the acidosis is coincidental caused by this. It's not essentially the lactate. So again, you'll always hear that people are like, oh, the lactic acid buildup, it burns. You do have lactic acid buildup, but it's not the thing causing the pain. It's actually, and it's not, it has a negative connotation, but it's actually not that bad. So we talked about anaerobic glycolysis. So this is actually this happening with the lack of O2. So although glycolysis does not use oxygen, the rest of glucose oxidation does. Both glycolysis and the citric acid cycle run independent of oxygen, but citric acid cycle requires the NAD+ that comes from the electron transport chain, which requires O2 to run. So essentially, citric acid cycle needs NAD+, and essentially how we talked about in this pathway, we can't recycle this because this has to go to the citric acid cycle. And this is preferred because you get more ATP when you go into the electron transport chain compared to redoing glycolysis. So essentially, this NAD+ is preferred to go to the citric acid cycle when there's O2 present. And again, the way we get this NAD+ back is through the electron transport chain, which needs oxygen. So the only way to get this back without doing fermentation is go to the electron transport chain. So when oxygen is present, the body's obviously going to prefer that because we get more ATP. So if oxygen is unavailable, glucose oxidation will get stopped at glycolysis, and the citric acid cycle will be slowed down because there's not enough NAD+. The NAD+ needed for glycolysis to run will end up getting depleted as glycolysis takes place, and the production of lactate ensures regeneration of oxidized NAD+, that's very important, from the reduced NADH, which is generated during glycolysis. So again, aerobic glycolysis has nothing to do with oxygen availability rather the rate of speed through glycolysis and the need for NAD+ for glycolysis to keep up with ATP demand. So essentially, we talked about how glycolysis is very fast. And if glycolysis ends up going into the other pathway, continuing through oxidative phosphorylation or metabolism, it takes too long. So essentially, the speed of what we need energy is faster, so it needs to go to the lactate so we can get back into glycolysis and repeat very fast. So again, here's a blood lactate and a workload in watts. So as our workload increases in watts, so this is likely a cycle ergometry or something like that, we can see blood lact can increase exponentially. Again, lactate dehydrogenase, Km, is less than pyruvate dehydrogenase. So essentially, lactate dehydrogenase has a lower threshold, so it will be active quicker. So again, here's our glucose. Our 2 ATP go in, we get our 2 ATP. We produce our NAD, 2 NADH. We have our 2 pyruvate, which become 2 lactate through lactate dehydrogenase, and then we get that NAD plus back for glycolysis. So pyruvate to lactate. So in this reaction, this is where we get that NAD plus back. So our substrates are pyruvate and our NADH from glycolysis. Our products are lactate and NAD plus. This is a hydrogenation or reduction. So here's a hydrogenation from pyruvate to lactate, and also a reduction through NAD plus. We require hydrogenase and reductase enzymes, and the name is lactate dehydrogenase, so LDH. You see a lot of these, and pyruvate dehydrogenase referenced in the literature quite a bit, as many different pathways that people study will involve these crucial steps. So again, the enzyme is named lactate dehydrogenase, since enzymes are always named for the reaction going in the direction of dehydrogenation rather than hydrogenation. So just keep that in mind. So again, the bottom line is that lactate is not bad. Lactate production is a benefit and can provide an advantage. So lactate production ensures regeneration of the acquired substrate NAD+, so glycolysis continue when ATP demand is high. So again, this is why when we're sprinting at that extremely high intensity 100-meter sprint all out, the intensity and energy demand is so high that we have to use this faster pathway, because we essentially don't have time or the resources to go do the citric acid cycle. So that's why when you're sprinting, you'll eventually you'll slow down, and you're slowly going to change over to the other pathways, and they're not able to keep up that high intensity. So that's why you're slowing down, at least from an energy standpoint. So when this happens, lactate actually results in little change to pH, or the acidity of the cell. Lactate dehydrogenase activity is greater than pyruvate dehydrogenase activity. This will mean there's more lactate at a high intensity of 30 to 90 max effort, so that 30 second to 90. Lactate can be converted back to glucose as a substrate for glycolysis. It's essentially as a form of gluconeogenesis. So essentially, this is when we form glucose through a novel way to generate glucose. So again, lactate dissociation is not the primary cause of fatigue during max exercise. It is the acidosis, which can result in approximately 10% reduction in force. And this is from those hydrogen ions we talked about. So cell-to-cell lactate shuttle. So there's mounting evidence that hypoxia, so that's low oxygen, is not the primary cause of lactic acid production, and that lactic acid could in fact be used as a fuel by numerous tissues, including both resting and contracting skeletal muscles. So again, it's actually can be a good thing. And again, it's not just this lack of oxygen. Oh, I'm tired because you don't have enough oxygen. It's different things. And the lactic acid production from this is kind of actually a metabolic adaptation to actually help us. So again, lactate is a valuable energy source. So we're exercising, our blood lactate so here's our pyruvate. They can either go to the citric acid cycle or lactate. So this is in the muscle. So this is a muscle. It's working hard, so we're going at a very high intensity, reduce lactate. So now this gets released into the blood, and now this blood lake can be taken up, converted to lactate, go from lactate back to pyruvate, back into glucose, and released glucose. And then same here, this pyruvate can then enter the citric acid cycle in other muscles. We can see here it can go back in the citric acid cycle in the heart, can be turned into pyruvate in the liver, either turned into glucose to be released or stored as glycogen depending on energy levels. So again, it has such a ton of valuable properties. So the pathways of lactate metabolism. So lactate moves from the cell into the blood via MCT 1 to 4. So this is essentially a lactate carrier. So lactate can be converted back to glucose through gluconeogenesis. So in the liver, gluconeogenesis is through the Cori cycle, or it can be converted to glycogen if it needs to be stored. If energy is, let's just say we restore our energy profile. So in the heart is a highly oxidative tissue with many mitochondria, and it uses lactate as an energy source and converts it back to pyruvate. Again, pyruvate can go back into its normal pathway into the citric acid cycle. It can go through the kidney to form gluconeogenesis, again through the Cori cycle. And lactate can go back into muscles. For example, slow twitch muscle fibers are well adapted to utilize lactate as an energy source. So lactic acidosis. So this is kind of what most people think about when they hear lactic acid. So in lactic acidosis, elevated concentration of lactate in the plasma, called lactic acidosis, occur when there is a collapse in the circulatory system, such as after a heart attack. There is a greatly reduced amount of oxygen available to the tissues, which causes cells to stop glucose oxidation at glycolysis, and this results in the buildup of lactate and a greater amount of blood lactate. The increase in lactate leads to acidification of the blood. So again, this is that crossroads we talked about. So essentially, pyruvate has so many different paths. So essentially, pyruvate can form alanine if alanine levels are too low. So essentially, we can detect. So we need essentially fixing it again. Same with oxaloacetate, and we need oxaloacetate to keep the citric acid cycle going. We need it to keep working. So if we're low on oxaloacetate, we can put it in. Then it merges with citrate synthase, becomes citrate, and then the citric acid cycle. It can also become acetyl-CoA in the citric acid cycle if aerobic glycolysis, so that's our O2 being present. And then it can also go to lactate in rest versus exercise, so again, that high intensity exercise. So we talked about the transport of pyruvate into the mitochondria. So now when we say, okay, our pyruvate is going to the citric acid cycle, this occurs in the mitochondria. So we need to get there. And not a lot of stuff can easily enter the mitochondria. It has to kind of use a lot of special things. So we'll talk about this carnine and as well as pyruvate. So when we talk about fat metabolism, but just keep that in mind. So we have a pyruvate carrier, which is essentially a transport protein. So you can see here's our respiratory chain. We have hydrogen going in, out and in. And we have a pyruvate going out and in as well. So the pyruvate dehydrogenase reaction is the link between glycolysis and the citric acid cycle. Again, always look at the number of carbons in reactant and products and what types of molecules or atoms are being removed or added. So here we have our 3-carbon pyruvate. Again, we have the enzyme CoA that's being added, and we have our NADH being formed again. Then we have the formation of our acetyl-CoA. We can see here, still 3-carbon, and then we produce 1 CO2. So, 2 carbons, sorry. So substrate, pyruvate, 3-carbon, NAD+, coenzyme A. So that's our coenzyme A right there, which is 21 carbons. Keep that in mind. The product is acetyl-CoA, NADH, CO2. The biochemical process is dehydrogenation, decarboxylation, and the enzyme is pyruvate dehydrogenase. So again, this occurs in the mitochondrial matrix. And again, when naming an enzyme that catalyzes more than one biochemical process, dehydrogenation will take precedence. So the pyruvate dehydrogenase reaction, again, is the link between glycolysis and the citric acid cycle. NAD is being added. This is the dehydrogenation. The hydrogen is removed from coenzyme A to NADH. So now we get that. And then we have the HS functional group, which is the thiol. So remember, NADH carries electrons that are transferred to the oxygen in the electron transfer chain. So when these NADHs are formed, they're going to go into the ETC and act as electron carriers. But there's a problem. So glycolysis occurs in the cytoplasm, but the electron transfer chain occurs in the mitochondria, and NADH can't get in. So essentially the solution to this is that electron carrying shuttles accept electrons from NADH. These include the glycerol phosphate shuttle and the malate aspartate shuttle. So these relate to they're using NADH there's both NADH and FADH, and these both result in different ATP yields, but these also enter at different, we'll see there's the complexes of the electron transfer chain, so we'll see there's 4 complexes, and different ones will enter at different stages, but we'll get to that in more detail a lot later. So the shuttles. Essentially, these shuttles act as entrance to the electron transfer chain, but this differs. Again, like I mentioned, the yield of ATP is different. So the glycerol phosphate shuttle, it yields 1.5 ATP per NADH. This is because FAD is an electron acceptor inside the mitochondria, and FADH yields less energy in the electron transfer chain. While the malate aspartate shuttle yields 2.5 ATP per NADH. So again, more ATP. This is because NAD+ is the electron acceptor inside the mitochondria, and NAD yields more energy in the electron transfer chain. Whoops. Summary. So again, the fate of pyruvate is that it's continuously converted to acetyl-CoA and lactate. So again, this is with oxygen. This would be no oxygen. And again, it also depends on rest versus exercise and stuff like that. Again, there's a misunderstanding about lactate. Lactate actually helps to maintain high rates of glycolysis by regenerating NAD+. For step 6, it can also be reserved back to pyruvate or glucose in a bunch of different pathways, the heart, muscle, liver, and kidney, through the Cori cycle. The movement of pyruvate into the mitochondria involves a pyruvate dehydrogenase reaction. And again, pyruvate is the central crossroad of metabolism. And the movement of NADH into the mitochondria involves 2 shuttle systems. Those are glycerol phosphate shuttle and a malate aspartate shuttle. So again, that was part 3 of glycolysis. So as you can see, we started to get close. But next, we're going to be looking at the citric acid cycle and a few parts on that, and then the electron transport chain. These will take up quite a few lectures, and we'll go into a little bit of detail, and again, try and relate it to some exercise type of stuff as we go through. See you next time.